Well, hello. It's really good to be here and to share with you some of the work that we're doing in my lab. Title of this presentation, as you can see on the screen, is From Black Holes to Broken Bones. And often this talk title confuses people because, what's the relationship between broken bones and black holes? But my research group actually is split at the moment roughly half and half between people working on optical coatings for gravitational wave detectors through to stem cell research and trying to generate bone graft technology in the lab. And so the purpose of this talk is hopefully to explain to you what the link is between the astrophysics and the biomedical. I'm the head of Department of Biomedical Engineering at the University of Strathclyde. Often people don't know where Strathclyde is. Strathclyde is not a city in Scotland, but it's a region within Scotland. And so this university actually sits within the city centre of Glasgow, which is the largest city in Scotland. So to get started, this was our kind of big paper that we published back in 2017. This is what my talk is, to explain to you how we got to the stage. So in 2017, we published this paper that showed that we can promote 3D osteogenesis. This basically just means a volume of bone-forming material from stem cells taken from adult patients. And we use a nano vibration technique to stimulate the stem cells to change into bone cells. And so I'll explain to you how we got to this stage. So in 2013, myself as a physicist working in gravitational waves and now Professor Matt Dalby at the University of Glasgow, we published the first paper on this technique. And so this was the first coverage by the BBC back in April 2013, where we got to explain to the public for the first time that we could do this nano vibration, which was nicknamed nanokicking, to promote new bone being grown in the lab. So that was the very first paper, and it was quite exciting to be part of that. But if we reverse all the way back, back in the 1960s, 70s, cell biology was an evolving kind of new field of study. And people at that time, or biologists at that time, didn't think that cells would respond individually to cues in their environment. So the idea was that an individual cell was basically stupid. It didn't really have the ability to respond by itself. You needed to have a collection of cells. Now, this image here is an early image of a cell responding to its environment. So here, the cytoskeleton, so the fibers which hold the cell together, are stained in green, the nucleus in blue. So that gives you an idea of what these colors are representing. This is just a fluorescence imaging, so it's a clearer way of imaging the cell so that you don't have all the problems of hitting the diffraction limit of using a standard microscope. And here the cell is placed on a surface that has micron scale grooves. In actual fact, back when these experiments started in the 70s, some of the substrates which were used with these micro pattern surfaces actually were made by going down to the music shop and buying old LP records. And I think when I used to describe this 10 or 15 years ago, people weren't sure what records were, but I think they've become quite popular again. But they would take these music records and they would cut them up, and they would use the grooves that the needle would run along to play the sound. They would use these grooves to see how the cells responded. And this really surprised biologists at the time, because you can see here that the cell is aligning itself to these micron scale grooves. And it shows that cells individually can respond to their microenvironment. Now, the next bit of innovation happened between two professors in Glasgow back in the 1980s. Now, you might think that these conversations happen in the illustrious environment of the university, but actually this conversation happened in an allotment. So you can see a picture of the allotment at the bottom here. And Professor Chris Wilkinson was actually one of the fathers of electron beam lithography. So he was a physicist working in electrical engineering, trying to develop this method by which you could micro and eventually nano pattern surfaces in a very precise way. And Professor Adam Curtis was a biologist. He was appointed in the 1960s in Glasgow. Actually, he was the first professor of cell biology in Europe. And so that, as I said, emerging field of cell biology, he was actually the first professor to be specifically named a chair in cell biology. And they didn't actually hugely like each other, but they had this conversation in the allotment where Chris Wilkinson said, well, what happens if you introduce your cells to my very, very neat, very well calibrated microenvironment? And Adam Curtis, as you can probably see in this picture here, he was quite an eccentric man, and he looks that in his appearance, of course, jumped immediately onto it and said, yes, let's try this. And again, cell biologists at the time, they didn't really think that cells were particularly intelligent, and they didn't think that cells would respond to nanoscale cues, so features in their environment which were much smaller than the cell itself. And here are three nice example pictures. Here is a human fibroblast cell, and you can see here it looks very clear that the cell is moving across the surface, stepping on these 100 nanometer high platforms as if they're stepping stones, and the cell is moving across those. And then even zooming in, you could probably see if you look very closely at this image, you can see there's little protrusions from the cell. And if you zoom in on those protrusions, you can see how they respond to even smaller features. And you can see here that these protrusions are pushing against these 160 nanometer high little nano columns. And so these are these kind of protein structures which come out from the cell. They probe the environment, and in this case you can see that they're choosing to push against these tiny nanoscale features. And then this image on the right hand side is in fact a cell that has moved across and come into contact with a change in surface. So you've moved from a planar surface to a surface that has nanopits. Now these nanopits are so small, depending on how good the image translates through into YouTube, you may barely be able to see the nano pattern on this side. But you can see that the cell reaches this boundary. It sells these filopodia protrusions across these nanoscale protrusions, senses that the surface on this side is different, and actually the cell in this image, if you were watching the video, you'd see the cell moving across and stopping at this boundary. So it's able to sense that there's something different with the surface. And so this was very, very clear evidence and surprising evidence to biologists that cells respond to the nanoscale features in their environment. Now, people started looking at how this happens, and so there were two very important results. One is what we've looked at already, which is that cells have these nanoscale features, these protein protrusions coming out from the cell membrane, which sense nanoscale features in the environment. But the other effect is a more dynamical effect, which is this video is exaggerated using chemicals. But if you look at the cell membrane, you can see that the cell membrane moves around, or what we would say is ruffles. And so there are these two mechanisms which biologists started to think, well, are these the mechanisms for which cells are seeing nanoscale features, whether it's the nanoscale proteins coming out from the cell membrane, or the cell membrane itself moving backwards and forwards at the nanoscale. Now, what Adam Curtis, this professor of cell biology that I was mentioning earlier, what he wondered was, well, if the cell has these nanoscale effects, particularly with the cell membrane, if the cell membrane is moving at the nanoscale and is sensing the environment at the nanoscale, then he wondered, well, is it possible to vibrate the environment that the cell is in to confuse this natural probing mechanism, this natural touching and feeling, or I think what he used to describe it as a tap dancing effect of the cells. And so his challenge to myself and Matt Dalby, who I showed a picture of earlier, was, could we create a dance floor which could manipulate, how these cells tap danced and how they sense their environment and perhaps change cell behavior. Now, this was the almost the very, very first setup which we constructed in the lab. Here you can see that there's some petri dishes. So these are just standard culture plates where we grow the cells in. And in a standard experimental physics kind of experiment, we just kind of glued these all together with epoxy between piezo ceramics. So you can see here the small piezo devices. You can see the wires running off. If you take a zoom out, you can see here that we have an array of petri dishes. We have the cell culture media where we grow the cells in. It's red color, so that's kind of convenient to be able to see in the dishes. And then we have all these wires hanging off, and we're able to plug these together, apply a voltage. If we apply voltage, the piezo devices would expand or contract, and then we could basically shake the cells, very precise nanoscale displacements. So that was the idea. And when Adam Curtis started coming up with this idea, he knew that laser interferometry was probably going to be the method which was best for measuring these nanoscale kicks, these nanoscale vibrations, which we apply to the cells. And he knew that there was a group in the physics department in Glasgow University who worked on laser interferometers for gravitational wave detectors. So he had sent an email across just asking if anybody would be willing to help. And thankfully, that email came to myself and a colleague. My colleague was on holiday, and so I immediately jumped on the bandwagon and said, yes, I'll help you with these experiments. And so that's the experiments which I just showed you. Now, laser interferometry, for those who don't know, we obviously use these to detect gravitational waves, which are these tiny distortions in the fabric of space. But laser interferometers are useful for measuring displacements and changes in displacement in many different applications, and not just for astrophysics. And laser interferometry operates by using a single frequency, single color laser. We then divide that laser beam in half with a beam splitter and send the two halves of the beam up two different arms. Now, when that light recombines, when it comes back, it combines in a particular way because light is a wave. And so you can see here in this video that there's the representation of the wave pattern of the light. And when those two beams combine and they come back out, if the peaks line up with the troughs as shown here, then the light cancels and you get no light coming out from the detector. But if the relative position of those two end mirrors that you can see here, if they move, it causes the wave pattern combination to then change. And then if those peaks line up, then you end up with constructive interference, as we call, and therefore you get a bright output. Now, this is really powerful, whether for gravitational wave astronomy or whether for shaking stem cells at the nanometer level. It's really powerful because you're essentially using the wavelength of light to be your meter stick to measure your displacements. So it's a very well calibrated, very sensitive instrument. If you're thinking of the wavelength of light is going to be in some region of hundreds of nanometers, and therefore, you know, this pattern changing across is basically measuring some fraction of that hundreds of nanometers. And so very, very precise. Of course, people don't always think laser interferometers or interferometers in general are very easy to understand. This is a very ancient advert for Scottish whisky, which describes the first interferometer experiment, which is the Michelson-Morley experiment, which some of you may have heard of before. This was the experiment to detect the ether in space. And this advert for Bunnahabhain Scottish whisky says that the Michelson interferometer is as confusing as it sounds and even more complicated than it looks. Designed to detect the minute variations of light velocity through ether in space, it ended up proving that the ether was not there in the first place. Little wonder that the distillers in Bunnahabhain, 12-year-old single malt whisky, have no time for such scientific contraptions. This unique Islay malt defies any attempt to analyse its smooth, subtle qualities. Enjoying it is an art, not a science, and the only complication is in the name. If you've not tried Bunnahabhain whisky before, then and you like drinking whisky, then I would definitely recommend it. But definitely a difficult one to pronounce. But despite what people may think about laser interferometers, it was a very, very straightforward way for us to calibrate how we were shaking the cells. And so we were able to shine lasers off the petri dishes that were growing the cells in, and then calibrate, you know, how many nanometers, so this is billionths of a meter, were we shaking the cells depending on what voltage of signal that we apply through these wires that you can see here. And so we were going to aim for somewhere between 20 and 30 nanometers. So you can see, okay, we apply roughly 15 volts oscillating voltage, and then we should get an oscillating displacement applied to the cells of between 20 and 30 nanometers. So we were off. But just to give you a sense of scale, because, you know, for gravitational wave detectors, we measure these tiny, tiny displacements. But actually, in comparison, how how large a displacement we're shaking these cells is astronomical, if I can use that word, compared to what we do in the astrophysics side. And so here's a picture of one of the stem cells and from one of the early experiments taken by Habib Nikukar, who was a medical pediatrician who was our first PhD student working on this project with us. Here he's again stained the cytoskeleton in green and the nucleus in red, so you can see the structure within the cell. And there's a 20 micron scale bar here, so 20 microns, 20 millionths of a meter. So if you consider that we're trying to shake the cells around 20 nanometers, this is 20 microns divided by 1,000. So it's 1,000 times smaller than that scale bar here. So these are tiny, tiny displacements, and we'll explain a little bit more about how that looks. But that's how we apply the vibration to the cells. Often people who haven't studied biology so much, which was the case for me when I started these experiments, there's some confusion as what do we mean by stem cells? And often people think of the ethical issues around embryonic stem cells. And these are not the stem cells that we were working with. Of course, embryonic stem cells, which would be described as totipotent stem cells. So these are the stem cells that can form any tissue type in our body. Obviously makes sense. These are the cells that we start off with at the first stages of life. They form our entire bodies. But when we become adults, we don't have these embryonic stem cells in our body, but we still have what we describe as adult stem cells. And here's a list of some of those adult stem cells. And so the ones which we typically are looking at within clinical applications would be hematopoietic stem cells, which are our blood and immunocell lines. So these are the stem cells that differentiate to blood and immunocells. We have neural stem cells, probably no surprise what they differentiate into. They form the inert cells of the nervous system. And mesenchymal stem cells, and they can form all the connective tissue in our body. So these are tissues like bone cartilage, tendon, muscle, and fat. Usually patients aren't crying out for the latter. And so these are called multipotent stem cells. They don't form all of the tissue types, but they form particular tissue types in our body. And so these stem cells can be harvested from adult patients. And if you're able to grow them and differentiate them into particular tissue types, then you could, in principle, turn that to a patient if they require it for surgery. Now, in the very early experiments, we were looking at frequencies which were quite low. So we were thinking about, what rate do we shake the cells? And so we thought, well, if we think about walking or the heartbeat, then, this is roughly around 1 hertz, so 1 cycle per second. And so we started the experiments at low frequency. And when we were looking at mesenchymal stem cells, so again, these are the cells that form the connective tissues in our body, we didn't see any response to the lower frequency. But Habib Nikukar, who's shown here, when we were working together, we started looking at higher frequencies. And when he reached 500 hertz and 1 kilohertz, so that would be 1,000 times per second shaking the cells, he started to see very strong changes in how the cells appeared under the microscope. I'm going to show you some pictures of how that looked. Now, this is what a mesenchymal stem cell looks like if you take out your human, out your body. So this is a cell that's been harvested from bone marrow. So this is a patient that's been in for a hip replacement joint. During that procedure, some of the bone marrow and bone is removed. The bone marrow can be scraped out. You can separate the cells which are inside the bone marrow. And one of these cells is the mesenchymal stem cell. So this is the cytoskeleton stained in white and the nucleus stained in blue. Now, again, being from an astronomy background, when I saw this picture, I just thought, okay, that cell looks like the Millennium Falcon. So that's like my calibration. This is what the stem cell looks like taken out from the body. Now, if you leave that cell just growing in the lab, it pretty much does nothing. It looks very similar. It will eventually form a fibroblast or an adipose cell. So it's kind of fibrous tissue or fatty tissue, which usually is not what patients are looking for. But if you shake the cell 20 nanometers, so this tiny 20 billionths of a meter displacement, 500 times per second, it looks like the Millennium Falcon to begin with. But you can see now that after 1 week or 2 weeks of this shaking, you can see that the cell has completely changed shape and it seems to be stretching out grabbing onto stuff on the surface and pulling, and you get this kind of what we call cytoskeletal tension inside the cell. And if you do it 1,000 times per second, in fact, the response was even stronger. Now, again, as someone from an astronomy background, I could just say, well, this looks like the Millennium Falcon and these don't. But when biologists saw this, they said, well, this looks exactly as you'd expect if the stem cell was differentiating into a bone cell. And this kind of makes sense because you can imagine that in the body, if that stem cell turns into a bone cell, it happens in a hard environment. Bone is the hardest tissue in our body. And so this is an environment where the cell can grab onto hard, rigid parts within the environment and pull on them. And so there's a physiological reason why bone cells typically look like this inside the human body. And for the first time, we were seeing this in the lab from a stem cell without the requirement for drugging, or what we would say using induction factors. I'm going to show you now the very short video which Professor Matt Dalby and I did for the BBC, and this was following the first publication of these results. And this was our first chance to communicate what we had found in the research lab to the public.
Stem cells, we all have them. They hold the key to repairing broken bodies.
Well, the stem cells that we use are taken from the bone marrow of adults. So they're within all of us, and the ones we're interested in can form bone or cartilage, ligament or tendon.
In this lab in Glasgow, they're turning stem cells into bone. There are already ways of encouraging stem cells to form bone. You can use expensive engineering techniques. You can douse them in a chemical cocktail. But here, they're going one better by giving the stem cells a kicking.
Well, in here we have the cells being nanokicked. Here in the petri dishes, which we normally grow cells in, the stem cells are growing. The piezo ceramics are attached underneath, kicking the cells 1,000 times a second at around about 20 nanometers.
So, not a very big kicking. We're talking billionths of a metre here, but it's enough to give stem cells the hint to start becoming bone. To get the nano-kicking just right, the team's using techniques developed for searching the vastness of space and time for gravity waves.
Here we're using lasers to try to measure the amount of kicking that we're giving to the stem cell cultures. So in actual fact, here we're using the wavelength of light to be our meter stick to measure the kicks down to nanoscale levels.
This is a stem cell in its original state. Give it a nano-kicking and the bone building begins.
If you take one cell and it blow it up to the size of a football, then the amount that we're shaking the cells is the same as sliding one sheet of paper in and out from the bottom. That's how much we're raising and lowering the cells as we shake them.
The research also involves Strathclyde University. It could help explain the ageing process and build new bones from a patient's own cells.
We're hoping to work more with people in the Southern General who work with patients who have had severe spinal cord injury, and again trying to use these techniques to perhaps try to regenerate the bone after an accident, for example a motorcycle accident.
So one day it may not just be cells, but entire patients who are given a nano-kicking. Kenneth MacDonald, reporting Scotland, Glasgow.
So that was the first BBC article, the first presentation of this research to the public. And very quickly, nanokicking, which is a made-up word, became a real word, at least to Google. And so if you search for nanokicking, you can see many, many pages. Most of them are referring to our research. There's some random things there. And then it ended up in newspapers, mainly in the UK, but there were some international coverage as well. And even in 2013, so the year that we published the paper, we even somehow managed to make it into a textbook, which was published at the very end of that year. And so there's a reference here to nanokicking, which was done by a team in the University of Glasgow. And so that was really good for us. But let me describe, what were the next stages which we had to go through to try to see if this was a useful technique that could be used for medical treatment, clinical treatments. So it's one thing to say, okay, we take the mesenchymal stem cell from a patient and we can shake this cell in the lab and we convert it into a bone cell. Now we want to know, well, do we convert it into a healthy bone cell? So what does a healthy bone cell do? A healthy bone cell secretes these minerals, these calcium and phosphate minerals, to make the hard ceramic structure that is our bone. And so the next set of experiments were were to see, well, if we leave the cells for long enough, can we see that they're behaving like healthy bone cells in the lab? So we stain here. This von Cossa staining is for calcium. This next staining is for phosphate. And so you can see here that if you take the stem cells from the patient, you just leave them in a petri dish growing for 34 days. You can see some very faint staining here to show that there's very, very minimal amounts of calcium and phosphate minerals being secreted by the cells. Now, these next panels here are actually for a near square array. So this is a nanotopography. So I'd mentioned before about how cells respond to the nano patterning, nanotopography in their environment. This was regarded at the time as the state of the art, again, a technology which was developed in Glasgow University. And there are many Nature papers publishing this work. And we grew the cells on this nanotopography. And you can see here that the calcium and phosphate mineralization is much stronger than in the control. But then we compared, well, what happens if you just grow the cells on a cheap plastic petri dish, standard culture plate, and you apply the nanokicking vibration, so the 1000 hertz, 20 or 30 nanometer amplitude. And here are the results here on the right-hand pane. You can see that the mineralization is really significant. So for us, this was a real breakthrough, that not only were we differentiating the stem cells into bone cells, but the bone cells were behaving like really healthy, active bone cells in the lab. Now, the reason why this was a big deal, and it was partly covered in that BBC article, was the fact that this might not have been the first time that this had been done in the lab, but it was the first time that it had been done without expensive engineering techniques. So this would be, for example, the nanotopography. It's a very, very expensive thing to manufacture. It's difficult to scale up. It's only on a 2D planar surface. The alternative way that people had been doing this before was to use chemical cocktails. So that's using induction factors, or you can just consider these like drug molecules to really chemically induce the cells to behave in a particular way. And a lot of those induction factors and drugs are quite harmful to reintroduce to the human body. So this was a real breakthrough because we were able to promote osteogenesis, bone formation, without the need for expensive engineering techniques and without the need for these kind of drug-related molecules. But the next thing is that we were still doing this in 2D. So we were growing cells on a surface and we were vibrating the cells on a surface. So then the question is, well, of course, a patient is not looking for a layer of cells. If a patient needs bone graft, they're looking for a three-dimensional volume of bone. So could you promote bone formation in a 3D construct? So then what we did was, well, we then modified our vibration platform to be a little bit more user-friendly. So this is the picture of how it looks now. And within a 6-well plate, so you can see there's 6 volumes in here, instead of placing the cells just with a liquid, we would disperse the cells in a gel. So a collagen gel, so a very soft gel where you could disperse the stem cells in a 3D matrix and then vibrate the whole gel and see, well, does this work? And without going into all the details, we again use various physics techniques to study what's the stiffness of the gel. These middle panes show computer simulations for how the gel was moving within the well that we were growing it in, these 6-well plates, so these kind of petri dish-like assemblies. If we shake the gel up and down, how does it move? And so we used finite element modeling. Again, it's a technique that we're very familiar with using within the gravitational wave research because we use it for modeling how our mirrors and suspensions behave and how quiet our mirrors are going to be in the astronomy observatories. And we apply this same technique to try and see, well, how do these gels vibrate? And then we use the interferometry again. There's lots of data here, but basically we did the interferometry for how the wells would behave and the gels would respond as a function of frequency, and around 1 kilohertz you can see down here. So in this region we have a fairly flat response, so basically the whole system just behaves in a nice repeatable way, that the whole system is just moving up and down 1,000 times per second. If you go up in frequency, then at some point you start hitting resonances, so this is where the system starts flapping around, but this is up above 2 kilohertz, so we don't really need to worry about that. So we went ahead, we did these experiments, we could see how these gels were behaving. So there's some images of these cells here. And then we did this von Cossa staining, so again, staining for the minerals which are being secreted from the cells. And then we were able to quantify, you know, if you just take the mesenchymal stem cells from the patient and don't do anything to them, this is our control. And then if you do the nanokicking, or if you use the induction factors, these drugs. So you can see here the nanokicking and using the induction factors, so these drug molecules, show a very strong increase in the mineralization. And then we were able to do some Raman spectroscopy. So this picks up various signatures depending on what atoms and how the atoms are bonded to each other. And then you can see here that the various peaks appear, and we know from the position of these peaks what material these correspond to. So again, you can see the phosphate and the calcium minerals, which are known to be components within the bone matrix. So again, here you can see a control sample against the nanokick sample, where you don't see these minerals appearing in the Raman profile in red here, and then you can see the minerals appearing when you apply the nanokicking. So this was really great for us. So this showed that you could apply this nanokicking and promote bone formation in a 3D gel, which then in principle could be reintroduced to a patient. If you look at our Nature paper, and I won't go into too much detail here, but there's lots of analysis for trying to work out, well, why do the cells respond in this way? And we found in all of these results that if you blocked two pathways, so that these are kind of biochemical pathways within the cell, so ROCK, which is associated with cell movement, and myosin, which is the protein, the motor protein actually causes the movement of the cytoskeleton fibers. So when you see videos of like cells moving across a surface, this is a myosin motor protein driven process. And so one of the things that we realized was if you inhibit those processes within the cell, so this is how the cell grabs onto surfaces, how it pulls on surfaces. If you stop that, then the osteogenesis, the bone formation, stops. And so this is really interesting, because basically what we've done is, if you have this cell which is in a really soft gel, you shake it with the nanokicking protocol, and actually you trick the cell into thinking that it's in a rigid environment like bone, and it's grabbing onto stuff and pulling. So basically what we've done is, through the vibration, we have done this manipulation of the cell behavior, where the cell thinks it's in a rigid environment and it thinks it can tug and pull on things, even though it technically can't. So that's some of the results, and for those who are interested, you can look up those papers. But then the question might be, well, you know, how can we use this for the benefit of mankind? And here is just an overview of the different areas where this technology could feed into. The first one, which is perhaps quite obvious, is it can be used in stem cell research and bone research. You know, we continue doing similar experiments in the lab. We understand more about how bone forms. We understand more about how bone cells secrete the mineral matrix. There's lots of stuff that can be done here. It can help understand the aging process, perhaps help understand bone conditions like osteoporosis. That's all fine. The final pain also probably makes sense, and that is, well, if you can generate bone precursor material in the lab, then you can reintroduce that to the patient. You can essentially use it like bone graft. The middle pain might be less obvious, and that is, you can use it for drug discovery. So if you're a big pharmaceutical company, you decide, okay, you want to develop a drug to treat a bone condition like osteoporosis, then you need to screen those drugs against cells. And if you're able to make bone cells behave like healthy bone cells in the lab, then it gives you a really accurate experiment, or more accurate experiment, to see, well, what happens if you introduce drugs to those healthy bone cells? How does it change those cells' behavior? And the really nice thing about that is that if you can make the cell-based experiments more accurate, then you reduce the number of animal experiments that you go to. And so this could have quite major implications for how the drug industry do the preclinical evaluation of drugs which are targeted at bone conditions. Maybe just to have a look at, you know, how you could generate bone graft with the current technology before nanokicking was discovered. You know, you have your human patient, you can take a stem cell biopsy from them. So that would either be extracting stem cells from the bone marrow, or in fact, mesenchymal stem cells are found in the adipose tissue. So you could do liposuction and extract the mesenchymal stem cells from the fatty tissue. You would use these reagents, so these drugs, induction factors that I've mentioned. You then make the stem cells or force them to differentiate into bone cells. You would disperse those bone cells in some kind of gel matrix, and that could be your bone graft. As I had said before, these drugs are quite expensive, but also they can be quite harmful to reintroduce to a patient. And so here's one example of how the induction factor, which is used at the moment for bone formation, is applied to patients. So one of the applications is for spinal fusion. You can see here a device that's placed between the two discs in the spine. The device looks something like this. So basically you have a holder, and inside that holder you have a gel, which is filled with BMP2, which is an induction factor for bone formation. And this was an article which came out in 2016. This technology is by Medtronic. And you can see here that it says here, while some of the records included good outcomes, and this is basically for spinal fusion, others showed more than 1,000 complications, some of which were linked to infuse, which is the material inside here. It ended up in 4 deaths, heart problems, and cancer. And so this is still a clinically approved drug, and it's still used in surgical applications around the world, but it's clearly not an ideal solution for patients because of all the off-target effects which can happen. And so our idea is, well, could you replace that with our Nanokick technology, and then you have a clean way to produce these bone cells and bone graft. And so again, these are the 3 areas which I've mentioned: research, drug discovery, and surgical bone graft. Now, just to give you a comparison for how our bioreactor technology against others. So, I mean, this is our platform here, really quite simple, vibration platform that you can grow your cells on. If you compare that to different perfusion and other bioreactor growth systems, then you can see that these are quite complicated. There's lots of pipes and all the rest. The risk of infection to your cells is quite high. It's very difficult to control. Our system, in principle, is very simple because you use standard culture where you don't have to have any automation kind of feeding of the cells, and you can basically grow them using standard protocols. I have a short video here which were made by one of our other PhD students that just shows how So that was our introduction, by which, sorry that the audio broke there momentarily when a phone call interrupted my computer. Anyway, often people asked us when we started publishing this work and seeing whether the technology could be used for clinical applications, they would say, well, this is great, but do other cells respond? And of course, it's kind of a hypothetical question that, do other cells respond to these nanoscale vibrations? And I guess some people have argued that the ability to sense pressure, which essentially derives our ability to hear and our ability for touch, our sense of touch, but the ability to sense pressure is the first sense which really evolved in nature. And so it might not be surprising that cells individually had the ability to sense vibration and sense pressure, and that this might be a whole avenue where cell behavior can be manipulated not by using chemicals, but by using mechanical cues or vibrations. And so here are just two examples, one of which is published in this Royal Society paper looking at bacteria. And what we did was, well, we stained for biofilm growth. So when you grow bacteria on a surface, they kind of ooze out this horrible material, which is usually the smelly stuff, which is referred to as the biofilm. And that biofilm serves many purposes. It actually also serves to protect the cells if you try and clean them. But we looked, well, if you apply the vibration, does it change the biofilm? And so we stained the biofilm and imaged it. And you can see that when we apply the nanokicking, the degree of fluorescence has decreased significantly, and so it looked like the amount of biofilm that was being formed had reduced, but you could also see structures forming. And so we published some of that result there. This was quite important because if the ability to control cell behavior using nanovibration is observed in bacteria, then that would likely confirm or back up the hypothesis that this ability that cells have to sense nanoscale vibration actually evolved very early on in nature, because you can trace it all the way back to bacterial cells. Here's an example which just gives really beautiful pictures. So I often use as an example where one of my PhD students, Christina Boyle, looked at the fusion in placental cells. And so this is another cell type that in the human body, a little bit like bone cells, they behave in a normal sense. So if placental cells fuse together and form the placental tissue. But if you take these cells out of a human patient and try to grow them in the lab, then they typically don't fuse together. And so people use chemicals and drugs to try to fuse the cells. But then, of course, those drugs can interfere with experiments which you're trying to do if you're trying to screen for diseases or other effects which are relevant for the placental tissue. And so we discovered that if you apply this 1000 hertz vibration to the placental lines, placental cells, they fuse together, and we got these beautiful pictures. So this was the first ever chemical-free fusion of these placental cell lines. Now, just to give you some idea of what we're doing, how are we helping to translate this to a clinical application to help patients? The Sir Bobby Charlton Foundation, now Sir Bobby is regarded as one of the most famous footballers. He used to play for Manchester United, a very well-known individual. He visited Cambodia many years ago and was very challenged by the number of people who were affected by landmines. And often when people step on a landmine, they end up having one of their legs amputated, but depending on the damage to the other leg will define whether the doctors are able to save that leg or whether that's amputated as well. And what the Sir Bobby Charlton Foundation did was to fund a large project that would basically take two bone technologies, one which is our nanokick stem cells, and the other one actually is a bone scaffold technology that can be 3D printed, and that's led by Manuel Simon Sanchez, who's actually shown in this picture here, who's head of biomedical engineering at the University of Glasgow, to take these two technologies where you could 3D print a scaffold, fill it with the nanokick stem cells, which we know are behaving like healthy bone cells, and reintroduce that to patients to replace large chunks of missing bone. And the idea is that you probably aren't able to save the leg that steps on the landmine, but if you can ensure that the other leg is saved, then that allows you to have the ability to fit a prosthetic to one leg, and you give mobility to the patient. And so for landmine survivors, this makes a massive difference in the outcomes and the quality of life that these people can have. And we have a new project funded by Horizon 2020 within Europe, and we're working with a cell manufacturer called Histocell that's based in Spain, who are going to try to manufacture the cells and then provide these for the first clinical trials, which will be in the next couple of years. We're also looking at trying to apply vibration directly to patients to reverse the effects of osteoporosis. Osteoporosis is known to be an effect. You can see here the difference between a healthy bone and an osteoporotic bone. In an osteoporotic bone, a lot of the bone material gets replaced by fatty areas. So these look like voids in X-ray scans, but actually this condition is nicknamed fat of the bone. And that's where the mesenchymal stem cell is differentiating into an adipose cell rather than an osteoblast, the bone building cell. And so because we know that our vibration protocol very strongly convinces stem cells to turn into bone cells, then we're applying these vibration devices to patients, in this case, patient with spinal injury, to try to reverse the bone loss that these patients suffer due to spinal injury and therefore lack of motion and forces and all the rest, mechanical forces on their legs and their bones. I will finish there, and I just put up this slide here because Mattis Riley, one of our collaborators in Glasgow University, on Twitter, if you follow him, you'll see that he does lots of these sketches to try to summarize people's presentations. So I once gave a talk where I actually highlighted the main results from LIGO and the first detection of gravitational waves, and then how we then moved on to working with Adam Curtis and Matt Dalby on these cell biology experiments, and then how we developed the Nanokicking platform from these kind of glued together Frankenstein wires hanging out devices through to kind of neat vibration plates that could be used by other labs and cell manufacturers around the world for the translation of this bone technology to the clinic. So I'll leave that slide up, and I'm going to take your questions now. So thank you for your attention. I see here that there are some questions. So Priyanka had asked a question, could you explain why shaking of these stem cells is required? As per my biology knowledge, when cells are grown in a petri dish, shaking is usually avoided. So that is true. I mean, in a lot of experiments, people try to avoid vibration, although usually vibration is not terribly important to cells. So cells are placed in incubators, which often have fans running, and there's various vibrations going on. And obviously within our own bodies, you know, we listen to music, we go to concerts, you know, vibration is quite a common part of our environment. In one of the early papers, which I didn't actually cite within this presentation, but when I started working with Adam Curtis, this question was very much on my mind. And so we did experiments where we would shake the cells at one frequency, and then we would track the frequency back and forward, sweep it between different frequencies, or we would like pulse the frequency. And so we did find that actually what's important here, it wasn't the exact frequency that you hit with the cell. What was important was that you stuck with that frequency. So it was as if the cells were aware that they were being shaken with a coherent, systematic, repeatable vibration at one frequency. And this was very important. And so we did those experiments. So for reasons which we still don't fully understand, but it seems to be very important that you pick a particular frequency and you stick to it, and then that gives the chance for the cell to respond in a particular way. Of course, it might be nice to know if other frequencies could form different tissue types. So I mean, if we have a vibration platform, it might be nice that we have a button not just for bone, but you could click for a different vibration protocol that might make muscle or cartilage or other materials. And we have some results relevant to that are coming out in papers which are being drafted just now. So hopefully that will expand our understanding of how this technique works. So I hope I answered that question. So yeah, why is shaking required? So the next question was from Rahul. How did you transition from biology to biotech? Do you still do astronomy? So I think I mentioned at the start that my research team is a team of around 10 people. We still work pretty much 50-50 between astrophysics and the stem cell technology. So we still do both, and I still work on both fields. And so yeah, for me, it makes life quite exciting. It certainly never gets boring because I get the privilege now of leading a department in biomedical engineering. So I see a lot of these really nice technologies that various people are developing for various things, whether it's medical devices or clinical therapies, and then translating them to the clinic. But I still work within the LIGO scientific collaboration. I'm the head of the optics, one of the co-chairs of the optics working group within LIGO. So myself and Jessica Steinlechner and Maastricht, we help coordinate the optics and the coatings research. So we're still very much involved. And I'm also very privileged to be involved within the Newton Baba project funded between the UK and India. And so I get to work with colleagues in India with the working of preparing and building for LIGO-India, which will build a third LIGO observatory, which will give us much better position sensitivity of where our astrophysical sources are on the sky. When we see the gravitational wave signal, how precisely we can locate which galaxy that came from. So it's still very much excited to be part of the astrophysics and working in the biotech. So yeah, we still do both. So there was another question about the frequencies. Have you tried different frequencies? Does it affect cell differentiation? So I had mentioned that it'd be wonderful to have buttons on our platform where you say bone, muscle, tendon, fat, whatever the patient needs. You press the button and you get it out. It's not clear to us yet that there's a magic frequency for different tissue types. We know that when we go to higher frequency, you get a mixture of effects. So you get some neural tissue, muscle tissue. Often the cells kind of complain a little bit and they start protecting the nucleus with the cytoskeleton. So there's odd things that happen at higher frequency. Whether there's anything that can be exploited there, I don't know. At lower frequency, we tend to just see the effect dropping off. What I can say is that we do have results in the pipeline that might be relevant for cartilage formation, but that's less to do with the vibration and more to do with some of the other conditions that the cells are experiencing as you shake them. So we've tried different frequencies. One of the challenges is that it's very easy, particularly for physicists like myself and probably many people listening to this. It's very easy to say, oh, we'll just choose a different frequency and we'll have a look. But the biology experiments are quite complicated, so they take some time to do. And often within biology experiments, they'll look for genes being turned on and off, which would be indicating that cells are responding in a particular way. But the issue is that when a cell, for example, a mesenchymal stem cell decides to turn into a bone cell, certain genes will be turned on at particular times and it will change the cell, but those genes will switch back off again, which means that it's not just the fact that it's difficult to see in the final result what you've done to the cell, but if you're trying to see if genes are being turned on and off, you have to look at the cell at the right time. Otherwise, it might just look like the genes are still off. But actually, it could have been that in between the times that you looked at the cell at the start and the end, various genes could be turned on and off in the cell. The cell could have completely changed what it's doing. But by the time you look at it later on, to you, it might just look like the cell's not done anything because all the genes have turned back off again. But the cell response has already been initiated. So these experiments take quite a long time to do. And so we don't know whether different frequencies really can be used to make the different tissue types and induce different cell responses. So I think I've answered the questions which I can see here on the list. So there was an anonymous question which says, what are your educational qualifications? So I started an undergraduate in physics and astronomy in 1998. In 2002, I started a PhD in physics, and I was working in gravitational wave, a gravitational wave group in the University of Glasgow called the Institute for Gravitational Research. So I was basically doing materials studies, particularly at cryogenic temperatures, relevant for the construction of the mirrors and the suspensions for gravitational wave detectors. From 2005 to 2008, I did a 3-year postdoc at the University of Glasgow, still working on astrophysics. I started during, then I moved on to a 5-year research fellowship from 2008. And in 2009 was actually the first time that I started speaking to Adam Curtis. And we started setting up the first experiments together. So that's kind of my career. So my undergraduate and my PhD were both really in physics and materials. The PhD was in materials, relevant for gravitational wave detectors. And it wasn't till 2009, so that would have been about 4 years into my kind of postdoctoral time, where I started working with the biologists. And so I guess for me, it's been a challenge to start learning some of the biology, having not done a biology degree. But one of the nice things with this collaboration is it is a collaboration of a multidisciplinary nature. And so we have the people who are real experts in cell biology, led by Professor Matt Dalby at Glasgow, the real experts in biomedical engineering techniques, led by Manuel Simón Sanchez. And what my team still bring is our expertise is in the precision measurements. So the precision measurement technologies which we use for gravitational wave detection, and we still try to develop these in relation to these stem cell studies. And so, although it might seem like my career has moved a lot in terms of the areas that I work in, actually the underlying technology has not changed much. And so both myself and the lead researcher, staff member in my lab who are working on the stem cell stuff, we both did our undergraduate degrees and our PhDs in physics and in gravitational wave detection. And so, yeah, the underlying technology is actually remarkably similar, although the applications between astrophysics and stem cells are very different. So let me just check if there's any other questions. So is it possible to apply different frequencies at different places in a scaffold so that cells differentiate into different types of tissue cells? So this is an excellent question, and I don't know if Siddhartha is asking for this reason, but you know, the holy grail for tissue engineering really is, you know, can you engineer organs? So, you know, can you actually engineer the different tissue types and then merge them together to make an organ? And that's like the holy grail in the future for this kind of technology. And of course, it'd be quite nice to know if there's different ways in which you can convert stem cells within a matrix so that you could actually make the different tissue layers or tissue constructs that might be relevant for patients and ultimately for parts or entire organs. I don't know how you can apply specific vibrations to different parts of a scaffold, because the challenge that you have is that the effective wavelength of 1 kilohertz in tissue, so in air it's many meters. I think even in the tissue in our body, it's still tens or of centimeters or still meters. I forget exactly what the number is, but it's large. And so, you know, when you think about these vibrations, because the wavelength is very large, it's very difficult, you know, to target small volumes of tissue with one frequency and one with another, because the frequencies that we're working at would just wash through all of the tissue construct or scaffold construct that you might have. So it'd be a real challenge to find a way to apply different frequencies to different parts of a scaffold growing cells, because those scaffolds are going to be at the kind of micron level structures, and the wavelengths of our acoustic frequencies that we're looking at are so much larger than those structures themselves. But it's a really good question. Can you apply different frequencies to different regions in a scaffold so that cells differentiate into different tissue types? It would be amazing if we could do that. It's not clear to me that the physics is making it easy for us. Let's just put it that way. So I think I've answered the questions I can see on my list. I hope that answers your questions. Okay, so I think I've answered all the questions. So thank you again so much for your time and for listening to this talk and your interest. Thank you for those that posted their questions, which were all really spot on in terms of, you know, the challenges we face with the technology. And hopefully you will hear good updates from the clinical trials that the Sir Bobby Charlton Foundation are funding, you know, in a couple of years' time. Hopefully there's some nice news that makes it out from that. So thank you, and we'll stop there.